New York Bio brings you Preparing for the Next Pandemic from our 2021 annual meeting, sponsored by Regeneron, featuring Florian Kramer, professor at the Icahn School of Medicine at Mount Sinai, Alina Baum, the director of infectious diseases at Regeneron, Carl Erlandson, who leads BARDA's SARS-CoV-2 Interagency Group Therapeutics team, and moderated by Mickey Atwal, vice president and head of molecular profiling and data science at Regeneron.
It's been 18 months since the pandemic has started. It still remains uncertain as to how it will further unfold. But what remains certain is that there will be a future pandemic at one point, one with potentially greater and more devastating consequences for human health and economic costs. So it is really important that we prepare for such a future event. So we want to discuss today what are the key lessons that we've learned over the past 18 months, how we've dealt with the pandemic, and what are the key challenges that we need to deal with to effectively deal with a future event.
I think in terms of science, both academia and also industry, people really ramped up quickly and addressed what they could, in terms of mitigating the effects of the pandemic, developing vaccines, developing therapeutics, developing diagnostics very, very quickly, right? We also have to take a step back and look at what happened. The official numbers are that approximately 5 million people globally died in this pandemic. About 6 billion doses of vaccine have been used 18 months in. But that means a lot of people have still no access to vaccines. And I think actually, while again, science did a great job, we need to get better prepared and we need to shut this down much, much quicker the next time.
I think, Mickey asked what would have happened 5 years ago. I often think what would have happened if this pandemic happened in the 1990s. And I think the global death toll would have been staggering. We would have been going through this much longer. We wouldn't have had the same capabilities to respond as quickly as we did with all the problems that happened, over the last couple of years. So I think the technology, the scientific discovery that piece by piece allowed the key players to respond so quickly, having the protein structure, having the sequencing data, having all of those things ready to go was actually incredibly instrumental and lucky for the whole world.
So one of the biggest points, I think, is just how much digital information is coming out. And this could be how easy it was for us to talk with other physicians in China or in Italy when the outbreaks were going on, or of course here, and really get to know the truth on the ground at the time that it was occurring and try to stay ahead of what's going on. I think also adding to that is, we'll probably touch on a lot, there's been a lot of very open science with data coming out rapidly. It could be sequences, it could be the bioRxiv publications where you see the early stuff. It could be Twitter. We've certainly been following that as well. It's been really useful. Also, as far as digital things go, we've had a little bit more luck with doing digital clinical trials and being able to follow up on people, and that's really useful when you want to keep people farther away and not have to bring them into the hospital or to the physician all the time to check for endpoints. And a lot of these things wouldn't have been as feasible only 10 years ago, probably.
Going forward.
I think the sheer amount of sequencing that has allowed us to track the virus during this pandemic is something that has never clearly happened before. And it's been fascinating to watch as the virus is evolving and changing in the human population. It's been also this data has been critical also for the US government making recommendations for treatments. They're now using sequencing data and really rapidly generated screening data in vitro to say, this therapeutic is recommended, this therapeutic is not recommended, and that's happening almost in real time. So again, I don't think this would have been possible even 10, 15 years ago.
Why do you think that is, and what needs to be done to overcome these constraints?
Those are basically regular barriers that you have. You need import permits, you need export permits, you need to have MTAs, and there are often no standard MTAs in place that can just be signed by a scientist, right?
It was our experience as well. In the beginning of the pandemic, when samples were precious. People were trying to get samples from individuals that recovered for COVID to isolate the antibodies. And all of those were controlled by government agencies overall. And we even, at Regeneron, we just went through a successful Ebola program. We clearly had the capability to develop antibody therapeutics for infectious diseases. We could not get access to samples for weeks.
We also had the same issues obtaining viruses from within the US in many cases due to MTA discussions from labs. Issues are concerning personal identifiable information. If you wanted to deep sequence the sample, I guess you could theoretically find out a lot about the person. And so we had a lot of complications just acquiring, not only internationally, which of course is an issue, but certainly also within the US, which is unfortunate.
We saw the first variant coming up in December, right? 2020 with B.1.1.7, and then B.1.351 came up next in South Africa, right? And those two countries, the UK, which detected B.1.1.7, and South Africa, which detected B.1.351, put a lot of effort into sequencing viruses that they actually isolated. And that's why they detected them. We did not have that system here. If you don't know what's going on globally, a variant might pop up that already dominates the scenario, infection scenario in one country, but we have no clue because there's no sequencing data available because there's just no sequencing technology or it's not large scale implemented, right? So I see that as a big problem, and I think we should make sure that these capabilities are there nationally, but they are also there internationally. And if a country doesn't have the capability to do that, I think we should help them to acquire that capability, because what happens somewhere in a small country across the globe might actually turn into a wave of variant infections in the US, right? So you always have to think globally when you think pandemic. It's not stopping at the border, that's the problem.
In fact, I think 80 to 90% of the sequences that we have access to are from the US, Europe, Japan, and that's it. And then everything else is a blind spot, as far as I'm concerned. And so if we can help to provide additional resources for sequencing efforts and personnel and ways to keep these machines running in these countries, I think that will be really beneficial now and certainly in the future.
From a scientific perspective, there's so much that's not known yet about the biology of the virus and why it causes severe disease. We don't know yet why it's so much worse in older individuals, why kids have these really mild infections. All these questions that were there a year and a half ago are still there. What is long COVID? What are we going to be dealing with, 5 years from now? We don't know. As a scientist, I think all of this is absolutely fascinating, and we're going to be learning a lot, but it's also clearly extremely important just for figuring out, how to deal with this moving forward.
There are, but I think the main problem that we have is we're not prepared and we don't have a system to be prepared, right? You can compare that a little bit to influenza. In 2009, I think the governments woke up and saw we're not doing very well. It takes us 6 months to get a vaccine, 6 months, much faster than now. I think, Carl, I don't know if you agree with me, but if worse comes to worse, we probably would have a flu vaccine available within 4 months of a flu pandemic now. And we just don't have a system like that for any other pathogen that's out there. We don't have that for coronaviruses, we don't have it for arenaviruses, we don't have it for hantaviruses, and we need systems like that. If we don't know what's going on, and if we don't have pre-pandemic vaccines that we actually test in phase 1, phase 2 trials, we will always be too late. And you can have mRNA technology as much as you want to. If you have to do a large phase 3 trial with prevention of disease as outcome, this will always take a long time. And I think we just need to put a lot of money into surveillance, preparing pre-pandemic vaccines and bringing them into the clinic, testing them and finding correlates of protection. And then I think we are in a situation where we can be maybe as fast as for influenza, where I think and that might be controversial, but if this would have been a flu pandemic, we would have been much better prepared. we have working antivirals, we would have vaccines very quickly. I think we just need to, and I'm not just talking about the US government, I'm talking globally. We need to put a lot of effort into looking for these viruses, characterizing them, and developing vaccines and therapeutics right away and not just waiting for the pandemic. These things have to be ready if one of these viruses jumps in humans and starts to take off. Otherwise, we'll always be too late.
So you clearly have to invest, and it has to be an effort across government and industry and academia. There's no way it can work otherwise. And you have to have these things ready to go, which is a huge effort, but hopefully we all learn this lesson of what happens when you don't do that, and that the cost, and we all agree, I think all of us, that there's going to be another pandemic. How quickly it's going to happen, what virus it's going to be, nobody knows. So we have to really invest in preparedness, and we also have to invest in manufacturing capacity. If you have material to run a clinical trial and you have something that looks great, how are you now going to make billions of doses to protect the world? That's another huge question that we're still figuring out.
An easier way might be to develop some products that have more cross-cutting effects, something that might work against flu and also coronavirus, for instance, something you could test right now in people and make sure that it works. And other alternatives that have come along regulatory-wise is to allow for testing of these products against diseases that are harder to find in nature in animals, and that's the animal rule, where they can develop the correlates of protection. These sound easy, but they are also extremely hard, probably on the order of running a phase 3 trial as far as time and money goes, because of the amount of model development that has to go. And then another thing I think that would help prepare significantly, again, is a cross-cutting type of approach, and that's going after host-based therapeutics, which target the host and the symptoms and maybe the over-aggressive immune response that some hosts might make to these novel pathogens. If we could develop something that targets, that doesn't target the virus, it would be less specific to a virus, could be utilized against a new virus as it comes out, but could also be utilized in day-to-day hospital treatment of people with ARDS, for instance, or other respiratory diseases.
And I think in the perfect world, we've been chasing these broad antivirals for a long time. The problem is, historically, the broader we tend to go, the less well they work. And I think it's definitely worth that effort, but it's an effort of, basic research, really basic discovery. On the other hand, we do have people may not realize, but what's been really important for us to learn is, for the first time, we showed that antibodies targeting the virus actually can have therapeutic efficacy against a respiratory virus. This has never been shown before. So we know that this approach works, and we know that this approach can actually be extremely fast. Broad antivirals are kind of a holy grail right now.
They've been the holy grail for a long time, and they're not here.
Which would be amazing, but much higher bar.
I was just thinking what's been the most useful right now, and what you saw at the beginning of this pandemic was hospitals being overrun, and so really trying to keep the hospitals from being overrun is a key priority. That can be due to increased surveillance, rapid testing that can be test to treat, having therapeutics available rapidly. So I think things like that would be really useful to work on in the future. Of course, I think we already touched on a lot of the other ideas on prevention for vaccines. I think certainly political messaging can be improved as well.
I think, to me, there's actually one lesson that was extremely valuable and very simple, and that's that masks work. They should be encouraged. The respiratory viruses are the biggest threat, and that's the biggest danger, and masks have been incredibly effective at limiting the spread, and that should be our go-to response on top of everything else. Hopefully we can all agree at least on that for the future, but probably not.
For me, I think what we learned in the last 18 months, we're not good at sharing, right? We don't like to give away vaccines to people who didn't get a chance to get vaccinated. Instead, we want to get vaccinated a third time, right? And so I think one of the key lessons is really we need vaccine production capacity globally, right? You need to have local vaccine production capacity. You need to enable countries to make their own vaccine so that they're not dependent on rich nations sharing their vaccines or rich companies selling vaccines to them, right? I think that's, for me, a really important lesson. If you want to battle or if you want to shut down a pandemic quickly, you need to have the capability to make a lot of vaccine locally and make sure that that also gets distributed locally, I think.
Thank you for joining us for Preventing the Next Pandemic from the 2021 annual meeting. Thank you to our panelists, and thank you to our colleagues at Regeneron for sponsoring this event. To learn more, follow us on Twitter at NewYorkBIO, check out our YouTube page, or log on to our website at www.newyorkbio.org.